progress in biology and medicine. Prior to UCSD, he was a postdoctoral scholar at the Genomics Institute at UC Santa Cruz and received his PhD studentship from Stanford University. His work has been conferred with the Best Paper Award at ASPLOS, IEE Micro Top Picks, and an NVIDIA Graduate Fellowship. Welcome, Yatish, and thank you for participating.
Thank you, Eric, for such a nice introduction and for inviting me to your esteemed seminar series. I'm particularly honored since I'm relatively new to phylogenetics research. I became interested in it around the time the pandemics had started. Eric, you are a veteran in the field, and I don't know what you feel about it, but I think it's a very significant period for phylogenetics research community currently. And that's because this pandemic has not only challenged the community in many unexpected ways, but we are also seeing phylogenetics research being applied in real world in several new and impactful applications. And I'm going to be talking about these applications in a bit. But before that, let me just lay down the context. I think since the pandemic has started, this visual that I'm showing on the slide on the right, I think is self-explanatory to everyone. So what this is showing is that as the COVID-19 virus called SARS-CoV-2 is spreading, it's also mutating, which means it's taking new forms. And some of these mutations are rendering the virus more contagious, virulent, or capable of evading the vaccines and antibody-based drug therapies. And this is the reason why scientists, since the very early days of the pandemic, have relied heavily on the use of genome sequencing that helps discover mutations of the virus and study its evolutionary dynamics. How heavily is this reliance, you may ask? That data has been shown in this chart. What it is showing is the number of SARS-CoV-2 whole genome sequences that are available on online databases through global sequencing efforts over time. Now note that this y-axis is on a logarithmic scale. Now to give you a context of how remarkable the scale of this data is, consider this. The database of another heavily sequenced virus, HIV, consists of approximately 23,000 sequences. Now prior to the pandemic, this database size would be considered large, and the tools capable of handling this scale would be termed large scale. But the SARS-CoV-2 virus has been sequenced 11.6 million times so far. And what that means is this is 500-fold greater than that of HIV database and is quite unprecedented, which is why our group has been referring to this scale as pandemic scale. And this is because we were unlikely to witness such a large scale sequencing effort outside the context of a pandemic. Now, another astonishing fact of this data, which I often share with my EC colleagues, is that during the peak months of the pandemic, this database grew exponentially from tens of thousands of sequences right here, which was already pretty large in size, to several millions. That's just in a span of a few months. So if you compare that to Moore's law, that's been shown in green, and it's to correct scale, it actually makes even Moore's law look flat. Relative to it, and that's because the slope of the red curve is 75-fold larger, 75-fold higher compared to this green one. So what this unprecedented level of sequencing data has done is to foster the emergence of powerful new applications in epidemiological, virological, and metagenomic applications during the pandemic. So to just give you an example, we are now naming lineages of this virus at an unprecedented scale and resolution and speed. So when I last checked, over 2,000 lineages of SARS-CoV-2 were named by the Pango group, which means that we are at an average naming over 2 lineages every single day during the course of the pandemic. Now, this is quite unlike the strain assignment that we've done for other pathogens in the past, which would remain unchanged for weeks, months, even years. In many cases. So note that the Pango nomenclature itself is phylogenetically derived. So for example, if you think of B117, that has been named as such because its subtree is nested within B11, which itself is nested within B1, and so on. So interestingly, some of these new lineages that we've been naming, such as Omicron, Omicron was, for example, named with the availability of only 4 sequences within days of its first emergence in a genome sequencing data. And this is why I think comprehensiveness of phylogenetic analysis is very critical to this particular application. And this is something that I will mention even later. So once we've identified these lineages, we can monitor how they are circulating at different scales, at local scale, at state, national, and global level, for example. So this data is now, in fact, being routinely shared in our mainstream, and that's because we want to keep our general public informed of the emerging trends. So for example, this chart that I've shown here has been taken from CDC's dashboard, and it is showing how the Delta variant, which has been represented in orange, became the dominant variant in the US between the months of June to September timeframe last year. And it wiped out the alpha and the gamma variants that were also circulating in high numbers in the initial months at that in this chart. And the same can be said of Omicron this year. When this variant appeared, it pretty much wiped out Delta and other variants, and now we are seeing that with Omicron subvariants. So now we are also sequencing at a scale that we are able to identify clusters of infection in a given geographic context that may seem those infections that may otherwise seem that they were unrelated. And we are also able to predict with reasonable accuracy where the infection could have originated from. So I'll give you an example. Here I'm showing a phylogenetic tree, and some of the leaves have been circled in red. And those represent a cluster of infections that were discovered in the state of Texas. And if you look at this phylogenetic tree, you would infer that the cluster would have originated in California, and that's because all the basal samples that you see to this red cluster were all found in California. So this process is in fact now automated and is helping public health bodies make timely and informed decisions for pandemic management. Another very cool application whose possibility I used to often wonder about as a graduate student is to be able to predict the fitness of an organism from raw sequence data. You can think of this as being like the ultimate application of computational genomics in some way. And it seems now that we are able to do this sort of prediction with decent accuracy for the SARS-CoV-2 virus, at least. This, you could say, is the magic of big data. Similarly, it seems that we are also able to predict the future dominant mutation in the virus based on its evolutionary history with acceptable accuracy. Again, I would say this is the magic of big data. Now we are routinely studying local outbreaks and super spreader events through genome sequencing data at an unprecedented speed, scale, and accuracy. Another thing that we've really seen for the first time something like this during the pandemic. Another of my favorite applications is that has recently emerged and has also attracted, in fact, quite a lot of attention from our global leadership. It was mentioned, for example, in the G7 summit as well. That is that of this wastewater surveillance. Now, you could think of this wastewater surveillance as being similar to the second application that I pointed out, except that the monitoring is happening using sewage data. So you don't have to even wait for an infected person to show symptoms, go to the clinic and get himself or herself sequenced before noticing the signs of the infection. And it can even happen at the level of residential buildings or offices or dormitories or nursing homes and so on. Now I should mention that UCSD is in fact a pioneer in this space, and the photo and the paper that I've cited or referenced here, they have been both taken from our campus. Now, besides the fact that these applications are really cool and fascinating, there's one more reason that I chose to highlight them. And that is because all these applications have benefited from the phylogenetic package called UShER that I'm going to talk about in this talk. And in fact, I think this package is the only one which is able to handle the scale and has enabled the phylogenetic analysis at a scale that is required for making most of these applications work. So hopefully that has motivated kind of the utility of this package. But let me now share, now in the next part of the talk, I'll share a few technical details of this package. So this package, we are calling it UShER, and the name is based on the first program that we released as part of this package, which was used to perform phylogenetic placement of samples. And that was published in Nature Genetics last year. Later, we also added the capability of doing phylogenetic tree optimization, so optimizing the tree structure, and then we added inferring recombination to it, and then we added another utility Actually, the order is not right, but yeah, matUtils was before both of these. But anyway, we added a utility to rapidly analyze the massively sized SARS-CoV-2 phylogenies using this package. So in the next part of this talk, I will go over details of each of these tools one by one. But before I do that, I would just like to acknowledge that the work that I'm going to present today was made possible through contributions from a large team of researchers and funding sources that I've mentioned here. And in particular, I would like to mention or acknowledge Russ Corbett-Datic, who is one of my mentors during my postdoctoral training at UCSC. And we continue to collaborate now very closely. And also Angie Heinrichs, who's a stellar scientist at UCSC. Russ, Angie, and I think, complement each other very well as a team. And that's because Russ has the ability to come up with really great ideas to work on. I'm usually the one to help with the implementation, and Angie is in fact the person who translates our work into real world impact. So for example, she integrated UshER into the UCSC genome browser, as well as the Pangolin tool, and made several other salient contributions. Of course, what I said earlier is a little more complicated, since Russ and I both have outstanding students and external collaborators who seem to be good at all three. That's idea, implementation, and impact. So I will be acknowledging individual contributions of our students in the future slides. All right, so now let me begin with the description of the first tool in our package called UshER that does phylogenetic placement of new samples. Now, in order to explain UshER, let me give you a real quick primer on phylogenetic analysis using genome sequence data. Let's use an earlier visual of the virus that's changing its form as it's spreading, shown here. We, however, don't get to see the spread, right? We don't get to see how the virus is spreading and how it's changing its form. And we also don't get to see these links, the transmission links, how the virus is spreading from one host to another. But what we get to see is the genome sequence of the virus after sequencing a sample collected from an infected individual. So imagine that A to F shown over here are different hosts. And the arrows between them indicate transmission links between them. And the color of the virus is indicating what variant that virus has, for example. So in the simplified figure, you could consider, let's say, A to be a reference sequence or the very first virus sequence that appeared in the human population, let's say. And as it spread from A to B, it might have acquired like this yellow mutation, right? And that resulted in a new form of this virus. Similarly, from B to E, it might have acquired another mutation, this green one, and so on. So the goal of doing phylogenetic analysis is to take this genome sequencing data, from which you can also infer which mutations exist in the virus, and infer the evolutionary tree. You could think of this as sort of the family tree of the virus. So looking at this tree, you can infer that B and D, for example, are more closely related to each other. They also share a mutation, yellow mutation. So they would be more closely related to each other than let's say to A and C. So this phylogenetic tree, if you look at it closely, then it kind of resembles, not exactly, but kind of resembles the transmission chain that has been shown on the left. So that's sort of one of the reasons why we like to do this sort of phylogenetic tree analysis. But the problem with this pandemic is that it's imposing a unique challenge on phylogenetic analysis. The data is not static, but constantly growing, as we had seen earlier. So for example, we now have over 11 million SARS-CoV-2 genome sequences available online, and tens of thousands of new sequences are being added to this database every single day. So how do you do phylogenetic analysis of SARS-CoV-2 with new sequences that are being constantly added? So well, one approach would be to do sort of naive de novo inference every time new sequences become available. So what this would look like is that you gather global sequencing data from databases such as GISAID, and you could already have a multiple sequence alignment of most of these sequences that were historically present, and then you have some new sequences that appeared. So what you could do is you could take those new sequences and add them to the multiple sequence alignment using tools such as MAFT, for example. And then once you have this MSA, you can do a de novo phylogenetic inference, let's say using tools such as IQ-tree. And so now you have these new sequences also being represented into the phylogenetic tree. Except the problem with this approach is that it's going to be intractable for the scale that we are dealing with, SARS-CoV-2 scale, which is in several millions, in fact, tens of millions now. So it's just not possible to do it, besides the fact that it's actually wasteful to do it, because you already knew the relationships between most of these past sequences already. So it's wasteful to do it again. So the second popular approach to do this would be to use what's known as phylogenetic placement, which means you actually take not just the new sequence data, but you also take the phylogenetic tree that you already knew for the past sequences that were available to you, and just place these new sequences onto that existing tree. So using a placement tool. And there are several options available, except what we noticed is most of the prior phylogenetic placement tools were also not not quite capable of handling the scale that we were seeing for SARS-CoV-2. So I'm just showing one of the placement tool data here. That's IQ-tree. To place 1,000 samples on a 100,000 sample phylogenetic tree would take several hours, besides hundreds of gigabytes of memory typically. And in fact, now the tree is 100-fold larger. So this is quite intractable with the past tools. So at that point, we decided to create, when we noticed this, we decided to create our own phylogenetic placement tool. We call it UShER, which stands for ultra-fast sample placement on existing trees. And as you can see over here, we were very fast compared to prior tools. For example, we were 3 orders of magnitude faster than IQ-tree. So what took hours could be done in seconds. Similarly, we were 1,000-fold more memory efficient. But even at some point, we realized that even this scale was insufficient for the COVID-19 data. At least a single server UShER, we realized may not be sufficient to handle the scale. So we had two amazing undergrads, Devika and Kyle. So Devika, what she did was to implement a utility called matUtils merge. With this utility, what you could do is you could parallelize the placement of independent samples on several servers independently on a single base tree. And then with this merge operation that she implemented, you could actually merge those trees back together to form a single giant phylogenetic tree. And Kyle managed to parallelize this whole process on the Google Cloud platform. And we saw very good scaling numbers as we increased parallelism, as you can see in the slide. So good, strong, and weak scaling. In fact, the loss of efficiency that you see over here is mostly because merge operation is using a reduction tree, and so there's not enough parallelism available, and this dominated the runtime. So, but as the tree size gets larger and as we place more samples, this actually becomes close to ideal. So you might be wondering, what's the secret sauce? Why is it that UShER is so fast? Especially when you compare to some highly popular software like IQ-tree, which as we all know, has amassed over 10,000 citations. It's not a trivial software. It's pretty well written and very well optimized. So what makes UShER so fast? So I could summarize the main factors behind a high speed up into these 4 bullet points. This is basically what led to the speed up. Number 1 was the choice of algorithm. So most of the prior placement tools were based on maximum likelihood, but UShER uses maximum parsimony, and I'll explain that in a bit more detail why we chose to do this. But that's one big factor. The other factors include use of efficient data structures. So for example, we invented a new data structure called mutation-annotated tree, something that I'll talk about again in a future slide. Then we did pre-processing of sequential placement that greatly helped. And we did efficient parallelization of the placement step itself. So I'll go over all these points one by one now. So the first one was the choice of the algorithm. Like I mentioned earlier, most prior tools use maximum likelihood. The problem with using maximum likelihood and the reason why it's so slow is because for every single site that you have in the alignment, you have to maintain a giant probability vector for all these bases. And as you can see, even to compute a likelihood for a given tree topology and a set of sequences, it's fairly computationally intensive based on this particular equation that I show here. But maximum parsimony tends to be much simpler. You actually can infer ancestral sequences, and you can infer exactly what mutations occurred. So no need to store these giant probability vectors, and also the equations are much simpler to handle. Now, most people think that maybe this is the reason why we have such big speed up, and that's it. But let me just mention that if you replace ML with MP, which is likelihood with parsimony, you only get 1 to 2 orders of magnitude speed up. That doesn't explain all the speed up that we got. We have to do other optimizations as well that I'll mention in the future slide. But another thing that people get worried about when we talk about maximum parsimony is that they are quick to dismiss that it's inaccurate, and so it's not very useful. But we decided to investigate whether that's actually the case or not. And what we found, what we concluded is that for SARS-CoV-2 data, maximum parsimony and maximum likelihood trees are practically the same. In fact, what's interesting is they are very highly correlated with each other. So what that means is, if you improve the parsimony, you improve the likelihood as well. If you improve the likelihood, you improve the parsimony score as well. So they are very highly correlated, and one of the reasons you could think of it, the reason being is this virus is very densely sampled. So when you look at the tree, you see very few branches which have a high number of mutations. So a very small fraction. It's very hard to find a branch which has 5 or more mutations, a single branch, because we're sampling it so densely. And so, in fact, what we are also noticing, and this could change very soon with some recent work that has emerged. But what we are also seeing is the maximum parsimony trees are better than some of the likelihood tools, even on likelihood scores. And the reason for that, we think, is because those tools are not exploring the optimization space. And I'll talk about re-optimization in a future slide, but they are not exploring the space as well as we do for maximum parsimony. This all analysis, by the way, has been done by Brian Thornlow, who recently moved to Rome Therapeutics, and he has a preprint on that too. And all the experiments have been put up on GitHub as well. But there's one more thing why I would just like to mention one more point about maximum parsimony. So based on the feedback that we've received so far, we've also realized that some of the users find maximum parsimony trees as being far more interpretable than maximum likelihood trees. And that's because maximum parsimony trees also annotate mutations rather than using probability vectors for different branches of the tree. It's therefore very straightforward to recover any ancestral sequence from the tree. And for many applications, it greatly helps simplify the downstream analysis and the math as well. So in the future, and this is my personal opinion, that we should be parsimonious about parsimony. What I mean by that is, if the maximum parsimony trees are reasonably accurate, I think we should prefer them over maximum likelihood trees. So don't add more complications than necessary. Anyway, so that was one factor behind our speed up, but there are other factors like I mentioned. So another one is what I call mutation-annotated tree. So typically what people would do with the placement tools is they would take a tree in UVic format, such as this, and they would also take in a giant multiple sequence alignment. So I've shown it for very few sequences. There are maybe a handful of sequences shown here. And SARS-CoV-2 has about 30,000 positions in its alignment. So that means that you would have several, maybe 100 kilobyte or so of data in the MSA itself. But what you really care about is how the mutations exist on the tree, essentially, how the sequences differ from one another in maximum parsimony. So what you could do is you could actually compress the data itself, which matters for maximum parsimony trees, by actually annotating mutations on the tree itself. So for example, in node 1, you just have 3 mutations, which are shown here. Node 2 has 1 mutation, S1 branch has 1 mutation, S2 has none, and so on. So when you do this for SARS-CoV-2, we know that the sequences are so similar. So most of the information in this MSC is anyway redundant. So when you compress it in this form, you actually it's not even lossy, and you manage to achieve a huge compression ratio, 1,000-fold compression ratio that I've shown here, and we call this type of compression as evolutionary compression, because it's sort of also representing how mutations have evolved over time. We've inferred it. We don't know exactly, but we've inferred it with reasonable confidence. So that's another factor. So having these efficient data structures means that we can load up even giant phylogenetic trees onto main memory, and sometimes this entire mat also fits within the cache. So you can do very high speed processing on this data. The third factor, like I mentioned before, is to do pre-processing of the sequential placement. So this is similar to the point that I raised before, where most tools, what they would do is they would take in the numeric file, they would also take in a giant MSA. So if you think of 3 million sequences of SARS-CoV-2, and I'll quote this number again and again in future slides as well, because we use this checkpoint for most of the performance analysis. Yeah. So this requires about 90 gigabytes of memory. So even to load that and then do parallel fit sync of which you would anyways do to infer where the mutations exist on the phylogenetic tree, it would take you 18 minutes on this tree to do this step. And that is required before doing the placement anyway. And by the way, this 18 minutes has been quoted for our tool, UshER. We have, and I think we have the fastest fit sync of implementation of any other tool actually. We parallelize it really well, but still it is quite a lot. And then you would take in a new sequence and place it onto this tree. So that there is a two-step process. But what you could have done is to just store this information itself, what we call the mat, and store it in a protobuf file, for example, in a pre-processed protobuf file, and that would do the evolutionary compression for you anyway. So you could represent this information in only 200 megabytes of data. And then all you have to do is just load it up and then do the placement, right? So just to load this mat, you would require about 20 seconds for this tree. So that means you actually attained a 50-fold speed up on this step, which would have been otherwise dominant in your placement. So you could just load it up and then do the placement, right? And then what you would get as output is another mat essentially, which you could just dump onto an output format. And then as you have more sequences to place, you can do it over and above this mat. So it's much simpler to do it this way than every time compute fetch and sync of assignments for your tree. So this is where we get another 50-fold speed up, in fact. So this is very important. And all you have to do now is to ensure that the placement of this sequence S7 is not going to dominate the runtime, which means it's much more shorter than the time to load the mat, which in this case is 20 seconds. So to do that, to ensure that this is not dominant, all you have to do is efficiently parallelize the placement step, and that was our last optimization. So I'll tell you how we do that. So you take in a giant mat and you take in new sequences, but you take the new sequence. You don't take in the entire sequence, rather you take in the set of mutations that sequence has with respect to the reference sequence that typically tends to be the root of this phylogeny. So you just compare and see what differences the sequence has. And then what you could do is you could do a brute force search in parallel at every single branch of the tree and ask the question, what would be the parsimony score to place that sample onto that branch, both as a sibling and as a child. And so this has been shown in this particular chart where we've parallelized the placement over every branch of the tree. And then you pick the one which has the lowest parsimony score addition. And in this case, that turns out to be node number 1. And you place the new sequence right there. So S7 is placed right over there. And then the output itself becomes a mat because you already knew which mutations you need to add to incorporate that sequence onto this mat. And so you just append that to the list of mutations that you have. So that's it. That's about the optimizations of UshER and how it works. And I'm going to switch to the new topic. I don't know, Eric, do you want me to take questions at this point?
People can always drop questions in the box, but I think,
okay.
Yeah, I mean, it's all been very clear so far. I love it. Thank you.
Okay. Thanks a lot. So let me move on. So based on what you just heard, you might be wondering, oh, we are doing some greedy sequential placement of samples. And if you've done phylogenetics and placement before, you know that this is not the best approach because it can sometimes lead to suboptimal trees occasionally. And what happens is over time, these suboptimalities tend to accumulate. So you made some guesses, and most of these suboptimalities, in fact, arise through ambiguous bases in the sequences. So you have to make certain assumptions about those ambiguities. And sometimes those assumptions are wrong. You make some greedy guess, and then the guess turns out to be wrong, but you did it once and you don't change it ever in the future. So you don't want this to happen. So the solution to that is to do phylogenetic tree optimization, where you do placement but you re-optimize the tree. So you explore whether you can change the tree structure in a way that you get better parsimony score. And this actually turns out to be very, very important, especially when we are doing placement of such large number of samples. So we realized we had to do this. So that's why we decided to implement this tool called matOptimize for doing that optimization step. So that's the point I'm making on the slide as well. The tree optimization programs help ameliorate the suboptimal placements. So what they tend to do is all optimization or tree optimization tools have something in common. Essentially, what they do is they explore tree rearrangement. There are various types of moves, NNI, SPR, TBR, all these different types. The example that I've shown here is an SPR move. And then they would follow 3-step process. The 2 steps I've shown on the parentheses are actually optional. But what they would first do is they would find if the tree rearrangement leads to a profitable outcome, which means in this case, for example, can I switch this particular branch and move it right here? And does that affect? Can I get a better parsimony as a result of it? And in this case, the answer is yes. The parsimony score changes from 12 to 10. And if that's the case, then you should keep that move. Some of the tools, what they would do is they would explore multiple moves in parallel and identify profitable moves, but some moves tend to be conflicting with each other, which means they cannot be simultaneously applied. So what you do is you apply through some heuristic only the non-conflicting moves, or using some sort of tie-breaking strategy for conflicts. Now I said that the first and the last steps are optional. So what some tools also do is they split up when the tree is very large. They actually split up the tree into multiple subtrees or sectors, and then they optimize each sector individually, and then they somehow have a way to merge the sectors back to an optimized full tree. All right. So yeah, and there are some tools which also what they do is they actually start with multiple copies of the tree, and then they explore moves and apply them in different orders, which means they are exploring different regions of the optimization space. And what they would at the end do is choose the most optimal tree at the end. So this requires, of course, this type of strategy requires a lot of memory. Anyway, another thing that we noticed is even tree optimization tools turned out to be insufficient for the scale that we were seeing for SARS-CoV-2 before. So again, we decided to implement our own tree optimizer. And the credit goes to my brilliant undergraduate student, Cheng Ye. What he did was to limit the space of tree moves to only SPR. We realized that it actually gives you very good trees at the end. So we limited only to SPR moves, and the flow chart is shown right here. What everything that we do, we find first the nodes to move, then we find SPR moves, profitable SPR moves. This step, then we apply those moves, and when we found that some moves we could not apply, which were conflicting with some other moves that we applied, then we recycle them, which means we will use them in the next iteration, and we keep iterating like this till we find that the tree cannot be improved further. The parsimony score of the tree cannot be improved further. And what we noticed is for big trees, for example, when you have million sample SARS-CoV-2 phylogenies, matOptimize actually is at least an order of magnitude, maybe up to 2 orders of magnitude faster, the best parsimony-based optimizer out there, TNT. And it actually gives you better parsimony score for if you have like runtime constraints. And in our case, we do have runtime constraints. We want to optimize the tree daily. So that's why we put a 24-hour constraint right here. And we see that the score is better for matOptimize. The improvement is higher for matOptimize in this case. I should mention that if you let TNT run for several days, it can exceed or exceed the improvement that you see for matOptimize, except that you cannot afford, because like I said, we wanted to do daily tree optimization. So and besides, now we actually have 10 million, so it's the scale is an order of magnitude, and I think TNT does not even work. At this point, it crashes typically, even if you provide it a terabyte of data. You can already see that with 1 million SARS-CoV-2 sample tree, it requires nearly a terabyte of main memory, which is very high. So it often crashes anyway. but yeah, we will try and improve this even further in future versions. But yeah, we are the more important thing to note is we are actually 1 to 2 orders of magnitude better even in terms of main memory usage. So you might be wondering what are the optimizations that we used in matOptimize, and that has been summarized right here. So yeah, unlike most other tree optimizations, we are based on the mutation-annotated tree. That gives us huge advantage because you don't have to load up the giant multiple sequence alignment, right? That I told you before. You don't have to recompute the Fitz-Sankoff states for that tree. That takes several minutes. Unlike some other tools such as TNT, we don't actually maintain multiple tree copies. We also don't keep the state assignments in recompute the state assignments. And unlike TNT, we don't explore the moves and apply them sequentially. So we have very efficiently parallelized it too. So what are the differences? We actually load up a mat, right? It's more efficient to do it that way. The loading takes just a few seconds. We maintain a single mat. This was just a design decision we made because we wanted to keep maintain a high memory efficiency. And then what we did was interesting parallelization where we parallelize the parallel move search, and then we identify the conflicting nodes and then only apply the non-conflicting moves at the very end. So that led to big speedups. There are some other optimizations in matOptimize as well. So for example, I mentioned before we use the mat format. Yeah, and the separate profitable move search was mostly done to enable this high parallelism. In fact, we also support multi-node parallelism using MPI. So you can now do this optimization on a cluster of servers as well if you want, and we are doing it anyway. And then we also did another optimization in the algorithmic sense. We actually modified the Gladstein's incremental update method to work with polytomies and calculate the parsimony score resulting from a single move really fast. And then we had all of this has been described in the paper or cited here. We also applied some novel search space pruning techniques that led to high speed up. But yeah, one thing I should mention is this efficient space of format that mat that I told you about. Actually, it's not the same mat that we use for UShER. You can see in the manuscript as well, but also here in this figure that we actually have some extra fields of data which were to support optimization. So they are more optimization friendly. So it's still a mat, but with some extra fields like fit set and boundary allele set and so on. And that just helps us explore the moves very fast. And you can read the details in the paper. Again, this scaling analysis has been done. Now this is old. Our scaling numbers are much better than what I'm showing here. But this is just to make the point that we have multi-node scaling available for matOptimize, and it seems to be scaling pretty well. And you can get your results in the order of minutes actually for even for very large trees, million sample trees. So this work, I should acknowledge, this matOptimize is not a trivial software at all. And what's amazing is it's actually been done by an undergrad researcher, and very deservingly, he actually won the department's best undergraduate research award this year. One last thing that I would mention before moving on to the next topic is that what UShER plus matOptimize is enabling us to do is what we are, or what people call online phylogenetics, which is different from de novo, which I shown earlier. So in de novo, you would every day take the samples and infer the phylogenetic tree, right? But what we do with online phylogenetics is we place the new samples on the previous day trees and optimize the tree structure with so placement with UShER and optimization with matOptimize, right? And most people would think that de novo inference has to be more accurate than doing this online phylogenetics approach, except that's we found that that's actually not the case. What we saw was, and this is what I've shown here is simulated data, but we see the same with real data as well. We tend to see that this online model of doing UShER plus matOptimize every day is actually producing better trees than other approaches, including the likelihood approaches. And this, of course, we could do it only for small trees.
So
Eric, did you have a question?
We did have a question come in from Yan Wang. So so Jan says that we've had problems using the Fitch algorithm on trees with polytomies because it doesn't always create the most parsimonious placement, and they've had to switch to Hartigan parsimony. Is that what you're using?
What is Hartigan parsimony? I actually don't know.
I don't actually know, but it sounds.
I don't know, but
So
no, go
he's.
ahead.
So I don't know if you know, but I mean, Jan also works in a similar sort of framework where they're using like a structure of a tree, actually trees, to compress genomic data. So he says this is for the evolutionary compression part of the pipeline. So Jan, well, maybe we'll just let Yatish go into the next part of his talk, and if you want to comment about a little bit more detail about Hartigan parsimony, we can talk about that.
Yeah, sure. I'll just mention that the FitSANCOV that we use in both UShER and matOptimize, they actually are sorry. So they can handle polytomies as well. So the mat we produce they all have polytomies in that, and the
Right.
algorithm is designed to handle polytomies. So it should be fine. I don't know if Yang has used UShER or matOptimize, but we do support it, and it seems to be working as far as we can tell.
Great, thanks.
Okay. All right, so that was about placement and optimization. I'm going to switch gears now slightly and actually talk about a biological topic. We all think of SARS-CoV-2 as mutating, and the sort of picture that I showed earlier, where it is acquiring single mutations at a time, and spreading as it's spreading. But actually, there's a spooky mechanism through which this virus also evolves. Most of you would be aware, but for those who don't, I'll actually give you a quick primer on that as well. And that's the mechanism is called recombination. And RIPPLES is the tool that I'm going to talk about next. That is actually to discover those recombinant sequences, and we have a phylogenomic approach to do this, and I'll talk about that next. So what is viral recombination? So sort of the mechanism has been shown here. Some people could be very unfortunate, and they could be infected by two different strains of the virus simultaneously. It can happen, and it has happened in this pandemic as well. So you could think of this as Omicron and Delta strains infecting the same individual. Could have acquired different strains or infected by different people and who were carrying different strains at the same time, let's say, or in close proximity. So what can happen when such a situation arises is that the virus can enter the same host cell, and you would know that the virus is actually using our host cell machinery to replicate itself. And during the replication, what could happen is, because both of these strains existed, you could have a phenomenon such as template switching occur, and what that would create is like a hybrid virus, which means that part of the virus is coming from one strain, or the new virus is coming from one strain, and part of it is coming from the other strain. So it has now two parents instead of a single one. So another way to visualize this is that you had two different recombinant parents that existed. They didn't look like each other. The circles are showing the mutations, and the line represents the genomic sequence. So you can see the mutations are very different in the two parents. But what could happen is that a template switching could occur at a breakpoint, and that has been highlighted by this dotted line. And as a result, you could produce a new recombinant virus, which looks before the breakpoint like the first parent, and after the breakpoint like the second parent. So this new recombinant is not exactly resembling either of the two parents, and it has acquired mutations from both of these parents. Now, why is this so special? And that's because, and why is it that scientists are so worried about it? And the reason for that is that, and this has been well studied actually in scientific literature, that recombination can actually lead to really drastic jumps in the virus fitness. So what I mean by that is, the new virus that got produced, the recombinant virus, as a result of recombination of E and F, it would actually take several steps, small steps, to produce that particular virus sequence had it done through the earlier mechanism which I showed, through acquiring single mutations one at a time. But with recombination, it can actually, what it can do is, okay, I'll acquire nice mutations from the first strain and another set of nice mutations from the other strain, and as a result, it is much more, it has much better fitness than either of the two strains, which were itself very fit, and as a result, it made a drastic jump. So this is a very scary situation. It can it can produce very dangerous strains very quickly. So that's why scientists have been worried about this recombination since the beginning of the pandemic and have been sounding alarms that recombination is going on. Except that at the beginning of the pandemic, a lot of those alarms were actually false alarms. But I could say that now I think the whole community has agreed that many of these recombinants are very much real. And for example, you might have heard the headlines of this Delta Cron variant that has emerged. That's pretty much real. Similarly, many Omicron recombinant strains have also been discovered recently. So now the question is, all these discoveries are, while they are happening, what we realize is much of this discovery is actually happening manually. That's you have experts who actually remember the mutations of individual strains. That's actually true. They've seen so many sequences, they actually remember it, which mutation each strain carries, and they've noticed a new sequence. Oh, this new sequence seems to have mutations from both of these strains, and then they look at it in more detail and they find that, oh, this is actually recombinant. There are some automated approaches as well, but they don't work at a large scale. So we decided that maybe something needs to be done about it, and we need to automate this process for really large scale that we are seeing for SARS-CoV-2. So that's why we created Ripples. And let me show you the performance numbers first before I show you what the algorithm does. We are actually 3 to 4 orders of magnitude faster than some other recombination tools out there like RDP, and we have much better memory requirements as well. And again, like for every other tool that I mentioned today, we actually did multi-node scaling for Ripples as well. So actually Ripples is very easy to parallelize, and that's why it has such good scaling numbers as well. So again, now you might ask, why are we so much faster? What makes Ripples so fast compared to tools such as RDP? And actually, that's been based on a phylogenomic insight, which actually Russ had. I'll mention what it does in a bit more detail. But let me first explain what other tools do, such as RDP. So what RDP-like tool would do is it would take in a set of sequences, for example, using multiple sequence alignment of all sequences that you want to analyze, and then it would actually try to choose triplets of sequences. So for example, it might choose these 3 sequences, 1 blue and 2 green. So it might ask the question, is the blue sequence looking like a hybrid of the other two green sequences? So that's what it would do. And it would do this for all triplets essentially. So it will also, essentially what it's doing is it's exploring all possible sequences as potential recombinants. That's 3 million of them. It's exploring all sites as breakpoint pairs, because you could have breakpoint anywhere in the sequences. So if you think of the SARS-CoV-2 genome, it would require billions such breakpoint pairs essentially, and then it would explore all possible potential parent pairs as well. So that's in trillions. So obviously this is not going to scale very well. Of course, we have search space pruning as well, but the pruning happens on this giant space. So it's just not tractable. So what we realized is, while we were placing these sequences which look like recombinants, they typically tended to have long branches. So what we did was, in Ripples, what we do is we start with long branches itself. So we are looking only for detectable recombination, where you can clearly see that they came from different strains. So when you look at long branches in the tree, there are actually very few of them, only 40,000 in a tree of 3 million sample trees. So it's a small fraction of the branches that are long, something that I shown earlier anyway in the maximum parsimony slide. And we restrict the breakpoint pairs as well to only mutation sites. So that also reduces the search space quite a lot. And then we restrict the potential parents that improve the partial placement parsimony. And that's because you want them to resemble the two parents, right, at the breakpoints. And we've already discussed, we have very fast placement tools. So we just reused it in Ripples essentially. So the parallel algorithm was reused. And we have a preprint on this already. It's currently under review. But with Ripples, we actually detected on a 1.6 sample phylogeny of SARS-CoV-2 over 600 recombination events. And an example has been shown right here, where these two strains have combined to form a new strain, and you can see how the sequences look like. Here's the breakpoint. Before the breakpoint, this recombinant in between looks more like the donor, and above and after the breakpoint, it looks like the sequence above, like this acceptor right here. And only the recombination informative sites have been marked here. So there are more mutations, but they could be shared. So to our knowledge, this is the largest recombination study that has taken place. Yeah, to our knowledge, this is the largest recombination study that has been done. And again, Cheng actually managed to optimize this even further. The runtime numbers that I've shown earlier have been improved, and now actually we can do 10 million SARS-CoV-2 phylogeny, the entire recombination analysis in under 2 hours actually. And so we've gotten faster. Anyway, one interesting insight that we discovered as a result of this analysis was that the recombination breakpoints are actually enriched or elevated in the SARS-CoV-2 spike protein region. And like I said, this work is under review, but we're pretty confident at this point that this seems to be the case, and this has been observed in some other viruses as well. All right, so I'm going to move on, and maybe this section would be quick. I'm going to talk a little bit about another tool, matOptimize. matUtils. That's actually a command line tool for rapidly analyzing the mutation-annotated trees that we share. So let me talk about this real quick. We realize that a lot of utilities for phylogenetic trees, they are also slow, but also they don't really work with mats, right? And mats are actually very fast to do analysis and querying on. So we decided to we decided to create a utility for mutation-annotated trees, and that's called matUtils, and it has various subcommands, and each of these subcommands actually have a lot of functionality, but I'm going to just highlight a few of these functionalities in more detail. So, for example, we added a functionality to annotate the mat itself with new clade names, right? So you could take in a mat, and just like we annotate mutations on individual branches of the tree, now you can actually annotate them with clade names. So here I'm showing, for example, next clade type naming strategy that has been used to add to annotate node 1 with clade 19A and 3 with 19B. So the output would be a mat again with clade names on it. Sorry, Eric, are you able to hear me?
Yep, everything's great.
Yes. Okay. Sorry. So I could not see my stream. Anyway, yeah. So that is one operation subcommand. And then similarly, this actually subcommand has lots of functionalities, but I'm just going to show you one. From the mat itself, you can extract out useful information that you care about. So for example, you could say that, okay, I care only about the clade 19B. Then can you give me a new mat which from this giant mat with only 19B as the clade output. So you could get a new mat out with just 19B. But you could actually extract, like I told you, has a lot of functionality. So you can extract a subtree based on mutations as well. You could extract it out based on branch lengths and several other conditions. So all of this, actually, you can see it on our wiki page if you're interested. Similarly, you can do uncertainty analysis. So for example, you can ask the question that, given a sample, how many equally parsimonious placements exist on the tree, which is sort of an uncertainty metric that we have. So you could generate a tabular output like this, which shows for each sample how many times, how many equally parsimonious placements. So B, you could say, is less certain than some other placements for other sample placements. Similarly, I mentioned the cluster tracker tool earlier, the California to Texas infection example that I had shown earlier. That actually analysis is done using matUtils introduce. So what you can do with this tool is you can specify the sample names which are found in a given geographic region. In this case, it's USA. So D, E, and F samples have been found in the US. And you want to identify in the tree whether this forms a cluster or not. And if it's a cluster, then which is the introduction node for that cluster. So that's what this particular output is showing you. So all this, there's a single cluster that's the monophyletic clade of D, E, and F. And the introduction is happening at node 3, which is what this algorithm has inferred in the simple example shown here. And then it will also tell you the mutation paths from the introduction node to that particular sample. And similarly, you can, if you want to extract out some basic statistics, then there's a matUtils summary subcommand for that. And like I mentioned to you before, this utility is actually very, very fast. On for example, 1 million samples SARS-CoV-2 phylogeny, you can compute the summary statistics in 5 seconds, extract out a subtree in 5 seconds again. You can extract out the mutation paths to every sample, from the root to every sample in the tree in about 15 seconds, and 9 seconds to resolve all polytomies that would otherwise take 37 minutes using another utility like ape. This utility has been developed by Russ's student Jacob. Introductions is also actually pretty fast. One minute to identify introductions in this large phylogeny. All right, so that's about our utilities. If Eric, you have 10 more minutes, I can actually talk about some of the real world impact that the tools have had.
That's it.
Okay. All right. So like I mentioned before, UshER has actually now been added to the UCSC Genome browser. So what users can do is so we actually maintain an updated phylogeny every day. So it keeps growing every day. And what users can do is they can upload their sequences onto this interface and get those sequences placed on the global phylogeny. And then you can also visualize that on the Genome browser. So you can see these are some of the samples that has been placed in this example. And you can also see the genotypes and compare them to other sequences out there. And also you can cross-reference with several other molecular and structural biology tracks that the Genome browser anyways provides. So all of this has been done by Angie, actually. She did this integration and has been very widely used actually during the pandemic. And if you want to learn more, if you've not used this interface and are interested in learning more about it, we actually have a YouTube tutorial. We have a playlist. UCSC created it, a playlist that goes over each of these tools in more details, and the link has been shown right at the bottom. So it seems like based on the tools we actually ended up creating, to our knowledge, by far the largest tree using genomic sequences for a single species, and we had a news release based on this last year, actually, when the size was about 1 million sequences in our tree, and we had an update once we crossed 10 million as well on another news release. Anyway, one more interesting thing happened during the pandemic is that people actually developed visualization tools. Not people, but actually Theo developed this really nice tool called Taxonium, that for the very first time helped us visualize our own trees as well. And now this tool has gotten really advanced. And you can see clades have been annotated on it as well, and they have been colored. And this is a very nice way. I think you can zoom in, zoom out, and see sequences. You can also search sequences that you care about from this giant phylogenetic tree. So this is really cool. But also UshER has been now integrated into the pangolin SARS-CoV-2 lineage assigner as well. So it was earlier in the version 3, if you use pangolin, it was actually a default. It was an optional mode. But later, I think we did some analysis, and it seems that UshER tended to be more accurate than using machine learning approaches. And so now it's actually the default. And the credit again for this integration goes to Angie, but we also thank the entire pangolin group for being open to this integration and welcoming it. UshER has also been now included in the CDC's genomic epidemiology toolkit. And so if you want to learn more about the tool, there's actually a nice module where Russ introduces how to use UshER on the Genome browser as well. But something that really inspired me was actually studies like this. So many of us read this headline that appeared in New York Times last year. This was actually the first study which had shown that with the Delta variant, children can get infected, and they can also infect others. Actually, the analysis of this study was done using UshER-based phylogenetic contact tracing that I think now has become quite common. In fact, Russ was involved in the study as well. And again, something that is very, very pleasing to see is that now UshER has been used for outbreak analysis in several parts of the world. That includes several low and middle income countries. Some of them are cited here. It was actually also used to flag, like I told you before, that Omicron was flagged with just 4 sequences that became available in our databases within days of its first emergence in the database. And that was done actually by Dr. Todd Peacock at Imperial College. And all such flagging is actually done on this GitHub repo. And he was the first to flag this Omicron variant with sequences from Botswana, South Africa, and so on. But what's more important is this plot where the UshER snapshot has been shown. So this is UshER, and we have auspice viewing in UshER now as well. So you could so it's been integrated. So this is showing essentially UshER-based phylogenetic analysis, and the first Omicron sequences have been highlighted in red. And as you can see, this is a really long branch, which means it has lots of mutations on this branch. And because several of these sequences, and they were relatively high quality, were present, we were confident that this branch is actually true. And what was surprising is so many mutations occurred at once on the Omicron branch. Anyway, we've also been downloaded, like on Biopanda, UshER is available, and that's actually a popular way through which our users are downloading UshER. And in fact, it has been downloaded over 150,000 times in a year and a half. We also had a lot of press coverage based on our work, and we are very thankful to the community for the sort of response that we've had for our tools. And I've shown some snapshots here. But yeah, with that, I would like to end this talk. And Eric, if I may, I would like to use your platform to also advertise that actually I have a new lab at UCSD, and we are looking forward to to hire students at all levels, undergraduate to postdoc. So if you're interested in analysis like this and are interested in sort of work that we do in high performance computing and compute architecture and so on, please feel free to reach out to me. So thank you.
Awesome. Yeah, congratulations on a really amazing body of work and a wonderful talk.
Thank you.
Let's see. So we have a question from Mustafa Ojanuba Jibrin. He says, are there particular properties of SARS-CoV-2 that enables the use of these platforms developed from your lab, or is it possible to use them for other pathogens?
I should have read that more carefully.
Oh, no. So basically, can we use this for other pathogens, or is it sort of a SARS-CoV-2 tool only?
Yeah, that's a very good question. I want to be careful about this. So I think this UshER-based analysis could work well for densely sampled pathogens where you don't have long branches and so on. I think there I think it could be reasonably accurate. We are in very early stages to investigate whether UshER works for some other pathogens. So for example, Russ is doing some work on tuberculosis data, and our initial results seem to be indicating that it's doing a reasonable job, but we need to do more work on this to be sure. But yeah, if you have dense sampling of any pathogen and you don't worry so much about long branch attraction, I think it could be fine.
Great. Two basically comments from Yan Wang. So he was just commenting the strict Fitch algorithm only works on binary trees. So maybe you're actually doing Hartigan parsimony without actually knowing it. But anyway,
yeah.
He also just and I think this is I also am thinking about this, but there's a lot of connections between what they're doing with TSKit and the mat work. You're aware of TSKit, and it's using a mat, but it sort of allows it to change as you move along the genome.
Correct. Yeah, I think they achieve very high compression, especially when you see recombination occurring in those sequences. So yeah, we are very much aware. We actually cite their work in most of our papers. It's a bit different, mostly because we are kind of also now moving, like our direction is a little separate in the sense that we are moving in more tree optimization and inference type of areas. So our data structures are a little more optimized towards that, and we are using a single static tree. That's the model that we are using, instead of them being able to model recombination events and more dynamic trees that way. So that in that sense we are different. We are thinking about how to combine these two ideas. We've been thinking about it as well. It's hard. That's what we realize. As soon as you start working with recombination and networks and multiple trees, the complexity increases very rapidly.
Yeah.
And you've been looking into it as well, Eric. So yeah.
Ripples sounds amazing, and it's awesome that you were able to identify all of these recombination events. And I'm sure many of them are authentic, but my guess is there's some of them that are false positives. And how do you estimate the false positive rate of ripples?
Yeah, very good question. So actually, so the 600 that I mentioned is actually post filtration. So Ripples throws out much more. I think we filtered it down to less than half that we think are more high confident recombination events. And we have a large number of techniques to do the filtration where we clearly identify that things were wrong. So for example, we often see problems with sequencing data, right? Where there is systematic issues that get created and they sort of start resembling recombination. We also see contamination in some cases that looks like recombination and so on. That's a little harder to deal with. But we have a large number of filters. It's all been mentioned in the there's a giant section on it in the methods of the Ripples preprint. So we do filtration. It's absolutely necessary. How did we measure the false positive rate? So we don't have great ways to do it, of course, with real data, right? But we did analysis with simulated data, and even there we see some false positives. We did some analysis with that, trying to model SARS-CoV-2 recombination using simulated trees and so on.
Great. Well, we're over time, so I'll stop there. Again, really wonderful talk, and thanks for kicking off our session on large-scale pathogen-associated phylogenetics.
Thanks, Eric, for inviting me.
Right, thanks. Have a good one.